As I mentioned in the beginning, we'll have two outstanding talks now and then a panel for the last 20 minutes or so. So now we'll continue with talk from a slightly different perspective. In other words, we talked about infections and antibiotic resistance. Right now we're moving to cancer and microbiome. So the first talk is about cell signaling modeling in cancer cells. So it is my pleasure to introduce the speaker of this for this talk, Diana Marculescu, who is a professor in the EC department at Carnegie Mellon University.
Thank you, Radu. And thank you all for being here. I know I'm between you and maybe an airline flight back. So I'm going to try to make it interesting for you. So just like Radu said, switching gears a little bit here, I'm going to talk about how we can model in a most efficient way cell signaling pathways, especially interconnected cell signaling pathways from the perspective of using hardware acceleration. So I'm going to start with a quote from Ray Kurzweil, who said that biology is a software process, but I'm going to change it a little bit in the sense that we can understand biology with Silico artifacts. And there's a lot of work at this conference on using computational means, especially in Silico simulation. This particular presentation looks at big mechanisms. So these are interactions and dependencies in cell signaling pathways. And typically these are available in technical papers, documents, or other written text. And typically they are extracted manually. So humans curate them and they come up with all these dependencies, they come up with hypotheses, and they try to make sense of them and test these hypotheses in the lab. The goal of this in general is to automatically extract such information via machine reading, usually natural language processing, and they would parse sentences like what you see there, and then come up with dependencies and causal dependencies or inference that can better inform how to build a model. These are qualitative in nature. So our premise is that quantitative information can inform this model better. So our goals are slightly different. We basically look at ways to augment this kind of big mechanism extraction with big data, meaning that we're looking at numerical data that exists in papers, supplementary information. They could be wet lab data or perhaps from other models, such that this qualitative information can be made more useful. And as a main goal is to also enable fast analysis of heterogeneous and communicating pathways. And we do so by using hardware. As Radu mentioned, the focus of this is to look at signaling pathways in cancer. And if you look at the hallmarks of cancer, most of the things that we are familiar with have to do with sensitivity to stress. For example, metabolic stress or mitotic stress, DNA damage stress, all these could be analyzed faster if you were able to do perturbations on various features of whatever model you're building. So one thing that we thought about is whether there is a way to convert typical nonlinear continuous dynamics that characterize these kinds of interactions to things that we know how to analyze and model in a fast and efficient manner. So for example, here on the left, you have something that could be converted in a sort of Boolean or pseudo-Boolean behavior that could be compiled directly to hardware. And by hardware, I don't mean programmable computing systems. I actually mean programmable hardware, which what this stands for is field programmable gate arrays. So these are specialized platforms that you can program to behave in certain ways depending on whatever input logical gate level netlist you introduce. So that's the idea. In other words, we come up with sort of an equivalent proxy, if you want, or an equivalent behavior in hardware that is typically much faster than the processes that happen in real life. So you could reason about outcomes much faster without having to go to the lab and try all the hypotheses that you have. So the focus here is on in silico acceleration for assembly and reasoning for this kind of signaling pathways. Some of the things that we need and we can work with include dynamical system simulation output. So any time series that is the output of a solver that you have, any kind of numerical data from tables, graphs, or wet lab data. As long as it's a time series or a time course, we can analyze it. And we can extract from there, either in a black box or a gray box fashion, a Boolean or a multi-valued or non-Boolean behavior, which could also have a pseudo-Boolean behavior, meaning that you can model non-ideal transitions between states. This could also be done once you have the model. You can do regular software simulation, but it lends itself to being very easily convertible to a hardware prototype if you want, in which case you emulate the behavior in hardware. It could also work with information that you have, for example, from a causal inference engine, in which case you can inform the model by using structure information. You can have partial models or new models, and then you combine it with whatever information you have. Once you perform the software or hardware emulation, you can generate hypotheses that could be tested in the lab and could go back to the causal inference engine to determine more dependencies. And finally, because it's much faster than simulation, you could use it to determine parameter selection that match the biological behavior. So for those of you who don't know much about the difference between simulation and emulation, I'm just going to give you just a very brief overview. In principle, simulation builds an artifact that behaves exactly like what you're trying to model. And usually when we say simulation, we have a software program that creates a model or representation of the system. So for example, if you have a protein-protein interaction network here, you can determine, for example, in this case, a logical model, and then you come up with a computer program that allows you to model the trajectory, dynamic trajectory of this model for various initial conditions and other types of perturbations that you're looking at. And this is what we typically understand through in silico modeling or analysis. Emulation, on the other hand, tries to imitate a certain system behavior. So it's a physical device that basically reproduces the behavior of the original system, and it has been originally developed by IBM. So it's usually done whenever we try to build new hardware or large-scale computing systems that we cannot afford to even simulate, because the difference between real-time operation and simulation is orders of magnitude difference, in which case you basically emulate the behavior using something like, as I mentioned, a free programmable gate array platform. So typically it's orders of magnitude faster than ordinary simulation, and because if we are able to find equivalent logical models up to some extent, right, to the dynamical behavior of the cell signaling networks, we can actually compile them directly to hardware. We can have instances of various types of, you can have a cell signaling for one type of cell interacting with another. So you can basically emulate these kinds of heterogeneous environments that we've heard actually in the morning that they're so important in bacterial infections, but they're also important in cancer. So now that we set the scene as to what exactly we mean by emulation, I am going to talk about two possible ways in which we can do this, coming from the time series. You take the time series or time course. You can actually determine these Boolean models or logical models using two mechanisms. One is a black box mechanism, in which case you do not have any domain knowledge, or you do not assume any domain knowledge. You can identify temporal dependencies, and then the resulting model could be used in a software simulation environment or hardware emulation environment, which is much faster. There's an alternative approach in which you can do gray box mechanism inference, in which case you do have access to some sort of domain knowledge. So you might know the type of mechanisms that are involved. You have information about the ODE dynamics, the network topology, or the interactions. So you can inform the mechanism inference gray box, and then you basically get a richer model at the end. And again, this can be done in the context of software simulation or hardware emulation, but also you can augment it with not just deterministic, but also probabilistic approaches as well. So now going to the black box approach. So here, the main thing I would like to emphasize is how fast we can come up with these models straight from the data. Typically, model learning from data can be pretty time-consuming, because what you actually need to do is come up with the best, in this case, Boolean model that best explains a bunch of data that you might have. And many times you have contradictions, and eliminating those contradictions may be impossible. So the way to think about it is how you can explain a lot of data with minimizing these contradictions. One thing we are exploiting are some of the dynamics that are in general very important and have biological significance. One is canalizing behavior, meaning that the abundance or absence of a certain protein triggers a certain outcome regardless of the presence or absence or anything else. And this is very much related to robustness, right? Because if you can control something by using one perturbation or perturbing one feature of your system, that is very powerful. And the second one is the fact that many of these mechanisms are characterized by a refractory delay or period, in which case you do have to account for the fact that some of these changes have to occur, or some of these abundance or absence of thereof have to happen for some time before you see any observable impact. In other words, explicit timing information has to be incorporated. It's not just you know, a simple time step. You have to look back perhaps multiple legs of a time. So both are essential for capturing correct biological behavior, but also it turns out that they allow us to do more efficient implementations for these. So for this black box implementation, you typically start with normalizing the data between low and high. You can binarize it. And that one way to do it is using k-means, and then you come up with a Boolean model that best explains the data by minimizing the contradictions between on and offset for any given variable or feature that you're looking at. And finally, what you come up with is a logical model that you can completely compile to hardware or just do, as usual, software simulation. It turns out that biology helps because canalizing features and refractory delays improve efficiency. So here you have just synthetic examples and two cases in which you do not consider refractory delays, or you do consider refractory delays. The cross-validation error for k-fold cross-validation actually converges faster in case of these refractory delays when they're considered. And intuitively, this makes sense because these trajectories end up in an attractor or steady state much faster. And a similar conclusion can be drawn when you use nested canalizing functions, in which case you have multiple features by which you canalize. So there's multiple triggers that allow you to get to steady state faster. Now, how does this work on actual biological examples? These are a few examples that are logical models in nature. They're from prior literature, from 19 features all the way to 47 or 49 features. Most of them are canalizing in nature. For the first one, all of them, and for the others, the majority are. But we also looked at dynamical models, executable dynamical models. In this case, both are executable in PySB, which allow you to do dynamic simulation using ODE solvers. So we wanted to see how well we can do for all these 5 models, how well we can determine these Boolean models, comparing with 2 state-of-the-art inference engines that allow you to best explain either in a best fashion or trying to find functions with no inconsistencies at 0 error. So both in runtime and error, our approach is orders of magnitude faster, and it can achieve trajectory fit. I have to mention that these are reduced trajectories, meaning that we remove all redundant transitions and dynamics. And then the 0% are actually achieved for a specific cross-validation setup. The kinds of things we can find are, for example, dependencies between cell death and the cleaved version of poly-ADPA ribose polymerase. And indeed, one increases with cell death. And in the other case, for example, the cytochrome C turns into the mitochondrial version turns into the cytosol version. And this is why one turns low while the other one goes high. So these are confirming existing knowledge. Now, how efficient can this be done? When you compile it directly to hardware, we looked at this in the context of pseudo-Boolean inference, in which case we don't necessarily look at only Boolean functions, but we try to sort of add non-ideal transitions with random variables that are fit here. So in our case, the pseudo-Boolean functions actually have coefficients that are generated in an interval 0, 1, so they're like random variables, in which case the transitions are not ideal, and they sort of can be as slow or as fast as these random variables are generated in a certain interval. The methodology is similar, except in this case we learn algebraic expressions that are converted to pseudo-Boolean expressions, and finally they end up being used in a hardware software emulation environment. Here's an example of some of the templates that we're using. So for various reaction forms, this is, for example, an inhibiting reaction or reversible, then you can come up with algebraic and pseudo-Boolean rules. And while you map them on existing ODs and reaction forms that you have, you can see how well they behave when you get your resulting pseudo-Boolean expressions. For the model 4, which has 36 species that we looked at, for a given trajectory, we're able to get very good agreement. And the kinds of things that you can do, for example, in the original solver, you would get something that looks like this. And with 3 orders of magnitude faster analysis, you could achieve the same steady state behavior. So instead of waiting, I know seconds is not a lot, but this is 3 orders of magnitude faster, which is much faster than the actual processes that happen there. So I'm going to conclude because I'm out of time. So data can augment qualitative big mechanism information that you extract from text, perhaps in an automatic fashion. And by using this hardware emulation approach, you can actually accelerate reasoning with these quantitative models 2 to 3 orders of magnitude faster than software simulation. Some of the challenges that we do have to deal with are incorporating or even learning structural information. So right now, in the black box approach, the kind of dependencies that you identify, they could be true dependencies or causal relations, or they could just be, due to the data that exists. And data can be noisy, in which case, of course, you need to incorporate stochastic behavior. So true logical models may not be expressive enough. So incorporating stochastic behavior would allow us to model cell populations or heterogeneous populations interacting. But the fact that we're able to compile them directly to hardware would allow us to do this analysis faster. And in hardware, we do know how to do interacting state machines and things like that. So this is something that we're looking at. With this, I would like to thank you and acknowledge my students and our funding.